when we do our experiment, the goal usually for the experiment is to obtain the true answer or true measurement. What do we mean by true? And how do we know that's a true answer, true value? So what do you think? So in our second experiment, we're going to measure the evaporation enthalpy and for the ethanol. And we are also going to measure the boiling point for ethanol at 1 atmosphere pressure. And once you have that measurement, when you have the results calculated, how do you know you have a true result? And the results should be correct. How do you know that? In the previous experiment you done for the labs, when you have a measurement, what do you do? What do you usually do? Compare accuracy and precision. But how do you know you're accurate though? What's the typical process you do? Well, I think you guys all did that before. It's just probably didn't reflect to you as a true value. So usually in other labs, I believe when you have a measurement, when you want to know if you got a correct measurement, professors will ask you, okay, can you look up the literature values and make a comparison, right? That sounds familiar. So I believe most of you guys, if not all, have some similar experience. So when you have to look up the literature values, that's the same for this lab. If you want to know if your measurement is a true value or not, I would ask you guys to compare that with a literature value. So once I have my measurement and there is a literature value, most of the time, you notice that most of the time, your measurement is not exactly the same as the literature value. Then how do you convince others that we have the true value? And how do we report those true values so others can understand that we have the true value? We're going to answer some of these questions. And in your report, I want you guys to present your data in a scientific way that other scientists would understand, and also would understand how close your measurement is to the true value. So first, let's review the precision and the accuracy. That those two concepts are related to the uncertainty, how well we know the experimental values. Precision is a measure of how closely individual measurements are agreed with one another, So between your measurement. So for example, let's look at these two pictures. The first one, I have all the darts on the center of the target. So that's pretty good accuracy and pretty good precision. And for the second one, all the darts are to the corner of the target. However, the darts are all together. That's pretty good precision. So when I compare my measurement with one another, I can say, okay, they all agree with one another pretty well. So that's good precision, even though sometimes the accuracy is not great. So for the third one, so accuracy here refers to how close measurements are agreed with the correct or true value. And in this lab, that means the accuracy refers to how close your measurement agree with the literature value. And for the third one, in this case, the darts are kind of away from the center. However, they are kind of around the center as well. If those are representing mathematical values, likely if you take an average, you can have the darts, the average value all in the center of the target. However, the precision here is quite poor because your measurements is all scattered. And what we want is good precision and good accuracy. We'll try to avoid those situations. But if that happens, we need to tell other scientists, how did that happen? And is my value in poor accuracy but a good precision, or poor precision, poor accuracy as well. Okay, so we'll see one example. So this is a scientific paper published last year, April, on the very prestigious biomedical journal. It's called the Lancet. And the title for this paper is Remdesivir in Adults with a Severe COVID-19, a randomized, double-blind, placebo-controlled, multi-center trial. So it's basically a medical trial to look at the remdesivir's effect on COVID-19 patients. So these two graphs are similar. Let's just look at the first one. A, the vertical axis here is the viral load. So how many the virus concentration we got from the patient. And the horizontal axis here is the time since the start of a study in terms of days. So from day 1 all the way to day 28. And the red line, that's remdesivir, and the triangle data point, blue triangle data point, that's the control, meaning there's no actual medicine that's given to those patients. The data points, the round points, the triangle points representing the average. You can see the arrow bar here that represents the uncertainty of each data point. Look at the day 1, there's no difference, right? Day 2, the viral load for remdesivir is even a little higher. But after that, that's approximately day 4. After approximately day 4, the viral load for patients treated with remdesivir is going lower than the control group, kept to be lower than the control group until after 21 days, until when the viral load is very low. So when the viral load is very low, when the measurement results is very low, usually there's a greater error in terms of measurement. Based on, if you are only based on the average of that value of the remdesivir group and the control group, probably some people can come to the conclusion that after 4 days, so this is approximately 4. So after 4 or 5 days, the remdesivir help to reduce the amount of the virus in human body, right? If you just look at the average value. However, in this paper, the conclusion is that there is no difference between these two lines. How do they draw to that conclusion? So they look at the precision of the data. They look at the error bar. So for this one, statistically, there is no difference between these two values because they're based on the 95% confident, these two values are overlapping here. Even though the overlap is a little smaller for this one, it's still overlapping. So statistically, those two data points are the same. And that applies to all other data points as well. So based on the statistics, there's no significant difference between the treated patient and the control group.